Hello everyone, I hope you have had a relaxing break after intensive discussions in the workshops. And here we are now in the Katrinenhof Research Center, let's say Necropsy room, with Dr. Dietmar Hummel. So he's going to lead this session, lead the necropsy, and first I would like to introduce him. So Dietmar is a veterinary by training. He has a PhD, and he is also a diplomate on the European Veterinary Parasitology College. He has been working here since 2012 as a senior clinical scientist, and he manages the clinical studies with large animals. He has been involved in the development of products like Eprinex, for example. So, Dietmar, before we start the necropsy, I would like to ask you to say a few words about Katrinenhof. So, what is it?
Yes. Katrinenhof has been founded nearly 45 years ago here in the south of Germany between Munich and Salzburg. And from the start on, it was basically involved in the development of antiparasitics. Mostly known is, for example, ivermectin and all the products which have been developed around it, as well as eprinomectin. But there's also a lot of work done in pet parasitics as well, and some work as well on antibiotics, for example. But the eprinomectin and ivermectin development is the largest development program which has been performed here over the years.
Fantastic. So we have 45 minutes for this session. So where are we going to start?
So we will start with a lung of a cattle, which is, we have collected this animal this morning, or the lung as well as the intestine we will work on later, from a local butcher. And we will have a look whether we will find any parasites, and we have additional samples which we will have a look after we worked on the lung, which are livers from cattle, sheep, as well as deer, to look for various liver flukes.
So let's get it on.
Right. So in cattle it's very easy, as we only have one lungworm, and we will try to have a look. So the lung itself doesn't look that bad in general, but the lungworms in cattle, which is Dictyocaulus viviparus, the name, dwells later on in the airways, and so if you want to have a diagnosis, you can open the airways, and usually it's easier when the lung is a bit colder, in order to have time for the worms to migrate into the airways, to the accessible airways, which you can cut open with a scissor. So we already started, and we opened the trachea, and overall it doesn't look that bad. There's not that much froth or mucus. And we will go on opening the trachea till the bifurcation, and we can have a look whether we see any worms.
So how big they can get, the Dictyocaulus? More or less.
Yeah, around up to 7 centimeters.
It's not that difficult to be found. I mean, at least when they are
they're
adults.
available. Yeah, so we need to check, but there's one, I think, or a part of it. And we place the nematode into physiological saline. They're very fragile. And if you place them only in water, for example, they will be destroyed. But this one is also already it's not complete. But we can have a look whether we will find further ones.
And they are always in the airways. They cannot be found any other place in the lungs?
Those stages we are looking for diagnostics, they are always in the airways, or also in the smaller airways in the bronchioli, which we will open in a minute. So, as for now, I don't see any further nematodes, and this is how you would open the larger airways first, and you see more mucus. But without any visible nematodes.
So, and when the animal is infected with Dictyocaulus, is there any necropsy finding, pathological finding that could be highlighted?
So you will find some more nematodes here.
Okay.
So this is close to the 6-7 centimeters, nearly. And in general, with the Dictyocaulus viviparus infection, we speak of a parasitic pneumonia, which will develop, and often with bacterial secondary infection. And depending on the initial infection, you have a more or less severe disease. This animal, in fact, as for now, as we didn't screen all the lung, doesn't have that much lungworms available.
Okay, so you know, with this level of infection,
Right.
you wouldn't see the animal coughing or any clinical sign?
Most likely not. So we can have a further look whether we will find some more nematodes, and you can easily open all the accessible airways with a small scissor with a knob in order not to destroy the tissue when you follow the airway. And this is in fact what we also do when we perform studies in order to assess the efficacy of a treatment. You would open the lungs of the treated animals as well as of the untreated animals in order to collect all the lungworms, to calculate efficacy and to produce efficacy data on that. But overall, in this animal, we don't have a severe picture of.
It's not highly infected.
No, it isn't. So I just switch the scissor in order to go here, but most likely It's not that easy to get a grip. And so usually they're still moving for some time.
So, Dietmar, there is a question here in the chat box. It's from Johannes. Do you also sometimes wash the lungs to rinse out the nematodes?
When we opened all the airways, we do a washing, but there's also another technique, which is retrograde perfusion of the lung, where you press water through the blood vessels in order to flush out the nematodes, which is quite time-consuming in a way. The lung gets very heavy, but the outcome isn't greater when you do these counts in this technique or whether you do the perfusion one. But afterwards, you can wash the lungs and collect the water in a Petri dish and screen a second time for those nematodes you didn't find at the first moment. It's more easy if you place the Petri dish then afterwards also on a black surface, as the nematodes are better visible then.
Yeah, it's interesting. We can see the Dictyoculus is still alive. It's moving.
It is.
Yeah.
But as the nematodes, or Dictyoculus viviparus is a very large nematode, it's easy to see, easy to identify, easy to collect, and easy to get the counts. This is different to the nematodes we will have a look later on in the gastrointestinal tract. So I think for now, this is how you would do the job in case you are interested in assessing whether the animal is infected with Dictyoculus viviparus and you get hold of the lung. You can open the airways and easily collect the available nematodes.
This is very good. So now, so before I have apparently there is another question in the chat box. So the first one is, what is the best way to diagnose lungworms or Dictyoculus besides necropsy and parasite identification? Can you perform fecal wag counts? Can you examine saliva or nasal discharge as well?
This is you can only do this test once. So this is a necropsy, and you usually need to kill the animal. But for the standard diagnosis, Beermann-Wetzel technique, larval migration is the most appropriate way to identify infection in animals.
Yeah, but I think, I mean, can you do fecal wag counts?
No, you don't do fecal egg counts. You do a larval assay, in fact. You do a larval migration from feces, and you collect them in, as the animals cannot swim, it's a standard technique. You place a bit of feces with some water in a funnel, and you can collect the larvae later on in a small glass vessel and screen the vessel whether there are larvae one available or not.
Yeah. And is it possible to find hypobiotic stages of Dictyoculus in the lung?
No, it's not the case in lungworms. So this is a classic development in gastrointestinal nematodes, but you will not find this in Dictyoculus viviparus.
Yeah, there is another question here. So was this a normal, healthy animal, not a clinical case, just slaughtered for human consumption?
So the local vet at the slaughterhouse screened it, and so far it was healthy, so it was good for human consumption.
Very good. No more questions here in the chat box, so we can move to the next.
Okay, I would suggest we will take the lung of the animal first and screen for fasciola, and later on we can have other livers to show all other liver flukes which are available here in Europe.
Great. So first the liver of the animal that was
right.
That was brought. Okay.
So this is the liver. Macroscopically, you can see it's a bit opaque, which is also typical. And if you take a close look, you also may see the bile ducts as well, which are a bit whitish, and you can see them on the surface of the liver as well. So for efficacy studies in cattle with fasciola, for example, you can also do an infection with fasciola, metacecaria, and depending on the stage you would screen for, you do the treatment, and you may collect afterwards, after slaughter as well, the flukes. For doing this, we usually cut the liver into slices and press each slice to push out the flukes, if available, and collect them in water.
Okay, so this liver has no sign of fasciola infection. Is there any?
It has signs. So okay. you usually the whitish areas are the areas where you can see the bile ducts, for example. The opacity of the capsule itself is an indication, but can also appear in other liver diseases.
So can you point where are those areas that you are mentioning? Please, yes.
So you may see them here, and if you push them, they are harder, so they are different to bile ducts you would usually not see, and if you cut them open, for example, and you would push them, they would be softer.
Okay.
But maybe we see also the flukes in here, hopefully, when we do the cutting. We can take off the gallbladder first. Haben wir da noch irgendwie was zum Auffangen? Das ist natürlich ein bisschen groß mit dem Messer. Das geht schon. And we can have a look at the gallbladder, whether we will find flukes in here as well.
So fasciola hepatica, right?
But so far, none are visible. So we start cutting the liver into slices. Around 1 centimeter. And after doing this, we usually also place them in a tub and fill it with water and leave it overnight in order to give them time to be soaked in water and repeat this process in the morning again. So this, for example, is, as we only see whitish areas or thickening. This is more typical for the chronic infection. And in acute cases, which you can see, for example, very nicely in sheep, as sheep are better host for fasciola in general, you could see blackish and reddish tracks of the migrating early stages of fasciola.
Which is the most pathogenic.
Right.
Yeah, that's where the pathogenicity, more serious pathogenicity comes from, right?
It's the migration, the destruction of tissue, the blood loss associated. But for now, maybe we cut it a bit faster. Soll ich dir die geben, dann kannst du nochmal vielleicht schauen. What you also see when you press the debris in the bile ducts. Hope that this liver is not a showstopper.
So probably while you are searching for adults or immature stages of fasciola, it would be interesting to talk about diagnostics. So how can we diagnose fasciola in a live animal, let's say?
In a live animal, so probably in cattle. For the clinics, usually it's not that visible as in sheep, while sheep has very typical symptoms. But in these animals, you may start with the fecal examination, which doesn't always allow the detection of eggs. You need for the for testing for fasciola eggs, you need another technique, which is called the sedimentation technique. So you just use feces and let the eggs sediment, and you can screen the sediment after staining for the golden fluke eggs. So they're yellow to golden color. And if you do the staining with a methylene blue, they are easy identifiable. There are other eggs available also, which you will find in the sedimentation, which is from rumen flukes, but they are not yellowish, they are clear, but similar in size. And
yeah, this is important because usually when a vet collects a sample and sends to the lab, if the vet doesn't ask for can you please look for fasciola eggs, probably what is going to be done is like a regular fecal white count McMaster, and nothing is going to be found, right? So if you suspect fasciola, you have to ask the lab to look for those eggs. So there is another question in the chat box. And it's Nigel is curious to understand why you leave the strips of liver on water or in water overnight, and what do you do the next day? Just more details about it.
So, in fact, we do the same, but the tissue will take up water and gets more brittle, and afterwards we do the same manipulation in order to press out the flukes out of the bile ducts. But for diagnostics, so you can also do serological tests. The ELISA is one test which you can do on milk, for example, which you can apply for herd screening, for example.
Yeah, we heard it all. We heard it about it yesterday in Johannes' talk. So another question here, Dietmar. So it's from Charlotte Harper. How reliable is the fecal egg sediment test to determine the presence of adult liver fluke?
Sorry, could you repeat this?
So how reliable is the sedimentation test to identify the number of adults in the adults of liver fluke or fascicular particle?
So if you find eggs, you can be sure that adults will be available. But in general, the test has its flaws, and not in each sample you will necessarily find fluke eggs. So as you have seen, the gallbladder is also a type of storage which allows the expulsion of eggs from time to time. So not necessarily your single sample will be positive. Yeah. But we did find one small fluke in the later screening, which is not the nicest specimen, in fact, and not the most typical specimen of liver fluke, but we can check in the lab to show us a nice one later on. So, unfortunately, that's parasitology. Not necessarily you will have massive amounts of flukes or parasites in general when you do a single screening in one animal. But nevertheless, we will move over to the sheep liver and hope that we will find dicrocolium in there. So these are from previous studies, and we have frozen some livers, which we can use for diagnostics here today.
Let's do it, because we have 15 minutes and
all right.
We still have the digestive system to explore.
I will need to fetch another dish. So for dicrocolium, it's even more important to collect the gallbladder, as the gallbladder is the place where you will find them. So let's see. So, we opened the gallbladder, and you can easily see there are structures in here, and all these around 5 to 6 millimeter large structures are Dicrocolium dendriticum, the small liver fluke as it is called. We can show them in parallel. Let's see whether this will work.
So the big one is fasciola, right?
Right.
From the Kathrinenhof collection.
Right, and they have a typical leaf shape, while these are way smaller and also called lancet flukes, as they are lancet shaped, and so they're easy to distinguish. But you can also find smaller versions, so the younger the flukes are, the smaller they are. And
yes, I'm just conscious of time. We have.
right.
Can we move on to the next
We
lever?
move on to the next lever.
I'm curious to see the deer one.
Up.
Oh no, it's okay. Just go. We can do it.
Die
later.
andere. So this is a deer liver, and this has been opened already. And this is Fascioloides magna, which is prevalent in North America, for example, and has been imported several decades ago to Europe.
And you can see why it is called magma.
Right. It is way larger, and while this inhabits the bile ducts, the gallbladder, this one, in fact, lives in the liver tissue, and that's why it's more damaging, and you will have more damage to the liver itself.
Much more pathogenic.
Right, and usually they live in large caverns.
Okay.
So I think
this liver is from.
this is a liver from red deer from the Bavarian Bohemian border, which is the area in Germany and also in Czech Republic where you will find Fascioloides magna.
So perhaps, Dietmar, we can move on
Yeah.
to the digestive system.
Right. So for next, we will start with the digestive system. We have already prepared the whole gastrointestinal tract with the abomasum, the small intestine, and the large intestine beginning here with the cecum. And we already prepared this, that we can do a quick washing of the material. So this is a standard procedure for collecting the nematodes. We will wash the whole contents of each separate organ in order to retrieve all the nematodes. And afterwards, if we have the time, we can show you how to differentiate them a bit and what would be the next steps. And so we will start with the abomasum first. Da wird ja nicht viel rauslaufen. Jetzt glaube ich, wir können es durchschneiden. Ein bisschen, schneid's mal. Dann nehmen wir die Kugel mit. So my colleague will support me with the processing. So we will first remove the abomasum, then the abomasum will be split open, washed with water, and the contents will be collected in a sieve down here. So we will start with the washing.
So Dietmar, a question from America Medeiros. Don't you tight the different parts of the intestine to avoid migration of the species as soon as the gastrointestinal tract is removed from the animal?
Sorry?
So if you tight the different parts of the intestine, usually it is done, right?
It is done for study purposes in order not to spread any nematodes during the processing, right? So you will start washing the whole organ and thoroughly wash the mucosa to remove all the contents. And then you will also wash it through the sieve. And once you have done this, you can first have a look for any pathological changes on the mucosa side and for collection of larvae. You can also do a soak, which means that you will transfer it into a saline solution overnight and would do the same process with a different sized sieve in the next day.
What sort of signs would you look for?
So we would look for any signs of bleeding, as this animal has been slaughtered in the morning. Maybe such bleedings are not that visible anymore, on the one hand, or for such nodules which are conspicuous for Ostertargia infection. So in cattle, Ostertargia is one of the main. And the main nematodes, in sheep it would be Haemonchus contortus. You will also find Haemonchus platzi in cattle, but probably the lesions from Ostertargia are more prevalent here, and we don't see any blood spots. But in Haemonchus you would see hundreds and hundreds of such red spots covering the mucosa.
So, and can we see those parasites with naked eyes?
With naked eyes, you would be possible for Haemonchus, which are visible very good, but for Ostertargia, they are way too small and very thin worms, which are not easy identifiable. So that's why we would thoroughly clean the contents and preserve it, and later on we would do a staining process and a destaining process in order to stain the nematodes and to differentiate them from plant material. And when you do this, you see the nematodes as tiny reddish worms. And while Haemonchus is visible by eye easily, you may see Ostertargia and you will not see Trichostrongylus axii, which also inhabits the abomasum.
So what are we doing now? We are washing the sieve? No, collecting this material?
Ich glaube aus dem Labor sonst. Aus dem Labor.
So why?
So we would collect this material. Fill this up with some water as well as with formalin for preservation, and then we could later on do the staining step.
Oh, so this is another question from America. So what is the size of the mesh you are using?
So this is a smaller mesh we have, which is 150 micrometers, and we will also use the same sized sieve for the small intestine. For the large intestine, you can go with 200 plus. So as the next step, we would start processing the small intestine. We do not need to process the whole one, but the first 3 meters should be sufficient to have a good oversight.
Yeah, and for clinical studies, you collect the material, then what do you do with it?
So all treated or untreated animals or groups would be processed in the same manner, and the material would be collected first for preservation, and over the time you would start screening the contents for the nematodes, but you do not need to screen the whole contents. You can do it on aliquots. So just a part, for example, 10%, 20%. And so my colleague will start to open the small intestine. And for the small intestine, the nematodes of interest are Cooperia species, and you may also find Trichostrongylus colubriformis, for example, Trichostrongylus longispecularis.
And any...
And
sorry,
in pasture animals, for example, also tapeworms.
Right. And any particular sign would you be looking for if you suspect parasitism?
We can move on cutting, but you would, for example, for Cooperia, which are regularly leaving their place where they take blood, so you would see also, apart from inflammation, tiny red dots from those blood-sucking nematodes, which are usually visible. So it depends on the age of the material you are screening. As this has been collected this morning, maybe this reddish color is gone over the period of time we... So I will start rinsing a bit. Sind hier etwa 3 Meter, oder?
Perhaps we don't need to do it all.
Yeah. So in fact, with this material you would do the same as for the abomasum. Collect it, preserve it, and then screen it also by staining the material. We can have a close look whether we will find any nematodes. As Cooperia can be visible, and that's looking good. Schwarze Schale, beziehungsweise. But you will not find Trichostrongylus. They're too small, way too small. So I would expect there's several hundreds in here.
Yeah, this animal was certainly not treated with Ivomec or Aprinex recently.
And also this species has probably the largest impact when concerning the small intestine nematodes.
Very good. Let's move to the large intestine. You know, we planned to end this session at 4:15, but we will end it a few minutes later.
So for this large intestine, we will take this. And we also will start opening it. And the nematodes of the large intestine, in fact, are easy to see with the eye. But there's quite an amount of material in here. This is the other sieve. So this is 300 micrometers. As the nematodes are larger, you can use a differently sized sieve. Vielleicht finden wir schon was.
So Dietmar, what nematodes can be found in the large intestine?
In the large intestine, we would look for Oesophagostomum radiatum in cattle and Schabertia, and you can also find Trichuris nematodes. But for a quick screening,
which one can you see with?
In fact, you can see all by eye. And you can also, for example, differentiate them if you're a bit used to it. But as it looks, there's one. But there's no Trichuris, for example, in it, which is easy recognizable as due to the thread and thin esophagus and the whip handle. Oh. No. So this is, in fact, very easy to assess any infection of the large intestine, but for the general use of such necropsies, it's good to have a general overview in animals which have recently or acutely died. You may take this approach in order to assess the infection with nematodes, which doesn't translate into this is the real cause of the death, not necessarily. But if you have large numbers of nematodes, this is probable. But that's it for here. And we can do a quick look at the... Was haben wir da für ein Organ?
So there is a question here from Gabriela Mancilla, I think. Did you recover the intestinal content under the table? Perhaps Sergio can show it under the table.
No, it's a runoff. It's a slurry. Yeah. No, we have a runoff here. So slurry pit.
You can show on your right, Sergio. There's a... Yeah, it's
Hemonchus, Ostertagia.
Was Hemonchus da? Männchen.
Ja.
Der Rest ist Ostertagia.
Yeah. Okay, we did a sample which is now stained, and we have Ostertagia species here, so thin worms which you would not recognize easily in the contents without staining.
So can you just separate the Ostertagia?
so the smaller ones, and we also have Hemianchus in this sample, for example.
So the Ostertagia is the smaller one, and the Hemianchus is the thicker
one, right. And Bunostomum. But usually, for example, for Hemianchus, you see it also in an unstained status very nicely.
Very good. So thank you very much, Dietmar. Thank you very much. It's always good to show the parasites in the animal, because one thing is when you're talking to a producer is to talk, parasites really have an impact on productivity. But it's hard for the producer to understand and to imagine the situation. But when you show the parasite itself in the animal, it's impossible for the producer not to be impacted, right? So this is one benefit of it. Very good. Thank you very much.
Thank you.
The skin, we can show perhaps we can show the skin.
So this will take 3 minutes probably. So we have from previous studies skin from Sauroptes infected animals, which we processed, and we can take one. So this is, place it just here. So Sauroptes is a mite infection in cattle, and these animals have been artificially infected in studies, and you can see on the one hand the clinical picture in a way in an untreated animal, where you have this thickening of the skin and you have small crater-like areas where you would also find in the live animal bacterial superinfection. This is quite smelly. You will have hairless areas, and you see the skin is very different to normal skin.
Rough. And also the consistency is completely different.
Right.
So this is not as easily manipulated like this one.
Right.
This is much softer.
This is much softer. And you will find this animal which has been treated, and after an observation period, which is usually 50 days plus, you have this picture. So you will it's still different to the normal unaffected area, but overall you see much better, and the healing in the animal is visible. So that's just an extra.
That's fantastic.
Thank you.
Thank you very much, Dietmar. With that, thank you very much, and thank everyone here in Katharienhof for the support. This has been sensational. With that, we'll hand over to Camilo. Camilo.
Thank you, Gustavo. Thank you, Dietmar, and for all the team here for this great demonstration of parasitological necropsy in a bovine. I think this is not every day that we can have this live streaming for such a practical session. So now we come to an end of this Global Ruminant Parasite Expert Academy, directly here from Katharienhof Research Center in beautiful Bavaria. We have so many thanks to give, but before that, also a great moment. Some of you, or many of you, are still waiting. We're still waiting. So we have our treasure hunt results. So I believe Serkan is sharing that the big winners of a wonderful earbud offer. We have a list of winners that were able to answer the 6 questions of the quiz, to answer them correctly in a fast way. And Serkan is going to share now in the screen the list of winners. Here we go. We have another one, 26 winners from all around the world. So we have all of you that are in this screen, check if you have your name there, you will receive an offer of an earbud. Thank you for participating and congratulations for the result in this treasure hunt. You got the name of the parasites right. So, moving to the closing remarks. Gustavo, I want you to join me here, please. You're the big maestro here. I will ask you maybe to make your final conclusions of this great technical workshop, and then we can move to the final thank you.
Yes, I think mission accomplished. I mean, thank you for the Katrinenhof staff, for our technical staff, for the supporting with the facilitation of the breakout sessions, the speakers, and in the first day we had those talks, presentations about diagnostics and resistance. Those were the foundation for the discussions on the breakout sessions, and now we are, finalizing this Parasite Expert Academy with this Necropsy, and fantastic. Thank you very much, everyone. And I hope next year we will be able to do this again, but now in a face-to-face system. And this hopefully COVID will give us a break. That's what I wish for. Camilo.
That's it. So we're now in the end of this session. Thank you again, everyone, for joining virtually. Again, as Gustavo said, as you can see, we hope to see you next year face-to-face. And thank you also for all the organizing committee, again, all the technical colleagues, all the staff here at Katrinenhof, all the parasitology experts that supported us here in this event. And see you soon. Thank you very much.